Hello everyone, we are presenting a new webinar series in our group RSG-Türkiye student group, and here our webinar is starting now. So for our social media, you can follow us on social media, and we would like to welcome you if you would like to join us and work with us. So here are our social media accounts. And today's presenters is Associated Professor Dr. Mustafa Tekpinar from Yuzuncu University, and his topic of presentation is Substitution and Deletion Effect Predictions with PRESCOTT. Now, the stage is yours, dear professor.
Okay, thanks for kind introduction. And let me start sharing my screen. Okay. You can see the screen, right?
Yes, hocam.
Okay. Okay, it's a pleasure to be here, really presenting my work and sharing it with my young colleagues like you guys. And thanks for your invitation. Today, I will talk about substitution and deletion effect predictions with PRESCOTT. And let's start here. Okay, I would like to start with a little bit of motivation. As you all know, amount of sequence data is increasing rapidly, but unfortunately, the amount of information and knowledge that we can extract from this data is not keeping up with the same pace. So we need to understand the meaning, biological function, etc., implications of the diversity in the sequences. And it's a huge, gigantic problem that everybody working on genes, proteins, genomics facing right now. So we need low-cost and fast methods to interpret the data. Unfortunately, typically, experimental methods are more expensive for this kind of analysis. So computational methods can provide a good venue for exploration of the genetic diversity. So the problem that I will talk about today is very simple, in fact, but unfortunately, the solution is not very simple. So let's assume that we have a short sequence of amino acids, starting with MDF, etc., and it goes on. And at the 8th position, one amino acid changes from Q to alanine or cysteine or D or any other letter, one of the 19 others. So this becomes single point substitution problem. Of course, it can be the mutations or substitutions can happen in any other places at the same time as well. If it happens only at one point, it becomes single point substitution problem. But if it happens in multiple positions, it's multiple point substitution problem. Similarly, some of the residues can be deleted due to external reasons or natural reasons, let's say. And if it is just one amino acid, for example, if Q is deleted at the 8th position, it becomes single point deletion problem. And if there are multiple deletions at various positions, it becomes multiple point deletion problem. As I said, the formulation of the problem is simple, but the solution is not that much. So the outline of my talk will be like a paper here. I will present the materials or the reference data that we are using, typical experimental or clinical data. And then I will introduce our method called PRESCOTT. Then I will share our results to show you the substitution effect prediction with PRESCOTT for single point and multiple point mutations. And finally, I will present my results on deletion effect predictions with ESCOTT method. Finally, I will introduce the software and the data availability so that if you want, you can also try with your own proteins of interest, the same method. So we have basically two data sets, and the first data set is called Protein Gym. Protein Gym contains experiments performed by various groups all around the world. And Protein Gym can be divided into subgroups, substitution dataset and the indel dataset, in fact. but we extracted here the deletions part only. So in the substitutions data set, we have 72 proteins from human, other eukaryotes, prokaryotes, and viruses. Various scientists performed 87 experiments over these 72 proteins. And if we divide it into two groups again, these experiments, 76 of them are single-point mutation experiments, and 11 of them are multiple-point mutation experiments. In total, we have about 1.5 million measurements of mutational effects. On the deletions data set in protein gene, we have about 60 experiments, again, in different organisms, and we have about 5,000 deletions, but we have some insertions, etc., but we are not investigating them here. The second major data set, or our reference data set, was ClinVar data on human proteins. In the data set that I will present today, we have about 2,000 proteins. We have about 8,000 pathogenic labels, and we have about 9,000 benign labels in this data set from ClinVar. So these are our reference data set that we compare our computational methods. So for prediction of variant effects, typically there are three sources of information. The first source of information is sequences. Typically, it's coming from an MSA file. The second source of information is coming from structures or PDB files. And finally, the third source of information is coming from population data. Here we are using human population data from NOMAD. It's a database from Broad Institute. So in our case, if we are using sequences and structural information only, we call our method PRESCOTT. But if we are incorporating population data also from NOMAD, we are calling it PRESCOTT. So I would like to introduce the basic ideas in PRESCOTT. Basically, we have two components in PRESCOTT. The first component is an independent component, and the second component is an epistatic component. We are summing them up linearly, but they do not have equal weights. We give 40% weight to independent component, and we give 60% weight to epistatic component. So, and our predicted effect, PE score of mutating amino acid X to Y equals to 40% independent component plus 60% epistatic component. So independent component is actually based on a simple idea. It's based on frequency of a certain amino acid in a column I or position I of an MSA file. The basic idea here is, okay, if in your MSA file you observe in the column that we are interested in, for example, first position, second position, etc., if the variant, if the amino acid is a lot, is frequent, it's mutating it to that amino acid will not be impactful. But sometimes the depth of multiple sequence alignment files cannot be too much, and you may have some limited data. Due to this reason, we are using an alphabet or basically a grouping. For example, we are putting methionine and valine in the same group, and the groupings are based on their physicochemical features. We are counting them and then dividing it by the total number of lines, basically. We convert it to a log score just for, let's say, further processing, but the basic idea is based on the frequency count in the multiple sequence alignment column. Before starting epistatic component, I want to introduce or pose a question, actually. Okay, how do we decide if a position in a protein is important or sensitive to mutations? For example, let's say we have a protein of 100 amino acids. Which position, third position, 15th position, is more important or more sensitive to amino acid mutations? The classical way is typically to take a look at the conservation, and then, okay, if a position is highly conserved, we say that, okay, this is important, it's sensitive to mutations. Here we are using a composite score, composite of 3 things. It's called max score. Max score has 2 parts. The first part is sum of TZ plus PC for that position I divided by 2, average of them basically. And the second component, TZ plus CV divided by 2. So whichever one of them is bigger, we select it. Here, TZ is an evolutionary conservation score. It is not Shannon entropy, absolutely. It's based on joint evolutionary trees, and for our research at least, it's a better metric of evolutionary conservation. The second part is physical-chemical features, and again, it's also like TZ between 0 and 1. And the third one, the third variable is CV, called circular variance. Basically, circular variance is a structural feature. It measures number of atoms in general, number of atoms around a position. So it's a kind of measure of a density around an amino acid or around an atom. So here, using max score, you can see that we are taking into account evolutionary conservation, physical-chemical features, and structural features. Now, we will use this max score to calculate an epistatic score. The epistatic score, let's assume that we have a query sequence that we wonder, okay, what happens? Can you see my cursor, by the way?
We can see.
We can see.
Yes.
Okay. So the top part is our query sequence. Let's assume that, for example, okay, we have V here, and we are wondering what's the effect of change to D. Now, in the MSA file, we try to calculate a score called evolutionary distance for each subject. And what we do basically, we sum up a score for all positions that are not identical to the query. For example, here in the subject, we are selecting only the red parts and then summing up max score of these positions for this subject, let's say subject 1. Since multiple sequence alignments contain several subject sequences, we basically calculate a matrix. For each line, you obtain a score. And it's called evolutionary distance matrix, Q, S1, S2, Sn, up to S, and whatever our uppercase N is, or number of sequences is. In the third part of the epistatic component calculations, okay, we are checking the subjects that contains, for example, D, all of the subjects that contains D at this position, as an example. And then we select the minimum value in the evolutionary distance matrix to calculate the epistatic component. And in this way, we are trying to see the epistatic effect of converting X to D or X to Y. Then we are coming back to our beginning point. As I said, we had 2 components, independent component plus 40 times 0.4 and epistatic component times 0.6. There is one final operation that we have to do. As I said, max score here is a metric of measuring importance of position. So due to this reason, we are multiplying our predicted effect with max score, and we obtain a normalized predicted effect for each mutation, X to Y. As I said, PRESCOTT has 3 information. All variant effect predictors have 3 information sources. If you are using sequence and structure, we called it PRESCOTT in our case. But if you want to add population information, how can you add it? The basic idea here is We are calculating ESCAT scores first. We are rank sorting them to produce numbers between 0 and 1. And then we are adding or subtracting basically in this equation, subtracting it or reducing it depending of its frequency. But when I say frequency, it's not amino acid frequency, it's population frequency. How many times do you observe it in a population? Luckily, there are very good databases like NOMAD that we know allele frequencies for several mutations. So the basic idea here, as I said, we reduce the ESCAT score in this way. We have a Heaviside step function shown with data. If f, the frequency of that allele, is greater than a cutoff frequency, it becomes effective. Otherwise, it's not effective. We have a C. The C is 1. We used it for tuning, but eventually we decided to use it as 1. And then we have a final component, FC minus F divided by FC, which is always between 0 and 1. So our cutoff frequency is 1 in 10,000, which means that, okay, if a variant is observed more than 1 in 10,000 people, you can reduce its ESCAT score, basically. Now the method part is over, and I want to present my results, our results for human proteins, especially in protein gym. I'm comparing it here with comparing ESCAT with 3 other methods. One is EVE, the other one is ESM1B, and finally it's the fourth one is AlphaMissense. All the other 3 are based on machine learning and deep learning models. ESM1B is a protein language model. And AlphaMissense is derived, or let's say, takes its roots from AlphaFold, basically. 2 things. First, our average Spearman correlation is almost identical to AlphaMissense score. Both of them are 0.460. And the second thing that we observe, you can see that for different proteins, the performance differs a lot. For example, for some proteins, almost all methods give more or less similar performance. But for some proteins, like SCM5A here at the bottom, almost all methods fail. So it basically means that there's a lot still to do in this field. And ESCAT performance is as good as AlphaMissense performance, despite the fact that we are not using any deep learning or any training. In the previous slide, I've just shown only 3 other methods, but here we wanted to put it in a larger scale. And here I am comparing our results with about 30 other methods. ESCAT and AlphaMissense, you can see, have the same performance or very similar performances. The third method is a derivative of ESCAT. Basically, instead of max score, we are not considering PC and CV, and we are using only evolutionary information. Despite this, IGEM score is also, the red bar is also quite good. And the purple one, or the violet color actually, the violet color shows GEM, predecessor of both ESCAT and IGEM. See, we are seeing here ESCAT performance is quite good for humans. And now here's a summary of them. I have already presented overall results. We wanted to divide our data set and to see if the performance will change for single point mutations and multiple point mutations. You can see that for single point mutations, our performance is 0.456. AlphaMissense performance is 0.456, and our performance is 451, which is quite close, but not the best one. On the other hand, when we check multiple point mutations, ESCAT predicts them way better. On the final line, we are not checking the variant effect, but we are checking, okay, which method predicts mutational sensitivity of a position. And you can see here PRESCOTT predicts mutational sensitivity of a position better than AlphaMissense. And even ESM1B are behind our method. Interestingly, I wanted to show you not just human species performance, I wanted to show you the other species performance as well. Here's the reason. Okay, AlphaMissense performs quite well when we consider other eukaryotes, prokaryotes, and viruses as well. And our method is the second one. But the dilemma here is this. AlphaMissense was developed to predict mutational effects in human, not other species. But despite this fact, they can predict other species as well, viruses especially. We don't know the exact reason why they are predicting, for example, viruses quite well. The unfortunate part for AlphaMissense is that their code is not public, and I do not know if they intend to make it public at all. Up to now, even though it's more than 1 year, they didn't release their code. PRESCOTT code is public with an open source license, and you can predict other species as well. Our predictions for viruses also are quite good, I could say at least. Now I want to show the CleanVar performance over 2,000 proteins, 17,000 variants. On the left-hand side, you see the distribution of scores for pathogenic variants, for PRESCOTT, and for ESCOTT. As you can see, and based on the idea I presented in the equation, you can see that benign mutations have much lower scores compared to ESCOTT here. Even ESM1B also have distinct distributions for pathogenic mutations and benign mutations. For AlphaMissense, you can see that it's not just on the benign side, but on the pathogenic side also, it kind of pushes the scores to two ends. When we calculate the AUC score for the Rockout curves, we can see that PRESCOTT score is 0.95. The second best is AlphaMissense, 0.91. And the third one is ESCOTT score. From here, we can deduce definitely that incorporating population information improves pathogenicity predictions here. Up to this point, the things that I presented are already in our bioRxiv paper, and we hope the main paper to be published in this month. From this point on, it is not published yet. It's an active research topic that we are still working on, but I still wanted to share it with you guys. So we thought, okay, we can predict substitution effects with ESCOTT and PRESCOTT, and what if we try the deletion effects? So in the deletion data set in protein gene, we have about 60 proteins here, 60 experiments, basically, not 60 proteins, sorry, 60 experiments, and we measure the performance of different methods. Here, the ESCOTT is the top row, and we have some protein language model-based methods like ProGen and RITA, and some other deep learning-based methods like transception or transceptive. We compare our methods. The results, if the result is dark red or dark blue, it means it's good, basically. Here we can see that ESCOTT performance is mainly red for many of them. But the interesting part is there are some experiments, some proteins that the predictions, like this one, TNKS2, the predictions are bad for all methods. And this is similar to the substitution problem. And here also we can see that majority of the methods are predicting quite bad, but ESCOTT performance is good. Now, this is for 60 experiments. What if we take the average of them for each row and then put it into a bar plot? When we do this, ESCOTT performance is 0.606, and it is the best compared to the other available methods in the literature. We wondered okay, if we can check the multiple point deletion effects as well. Unfortunately, for multiple point deletions, we have only 3 experiments, and 2 of them from human, and 1 of them is from mouse. And when we compare it, we can see that ESCOT predictions are good compared to other methods as well. So, but still, I mean, like, it's an important problem to note that, okay, we do not have many experiments that we can have a, I would say, robust conclusion for multiple point deletions. We plotted them as bar plot, the average of all methods, and for multiple point deletions as well, we can see that ESCOT performs quite well. So for the presentation of the method and results, I will stop it. As you have already seen, ESCOT predictions are quite good for humans especially, but ESCOT is not a method developed only for humans. It can be used for any species to predict variant effects in any species. And as I have just presented, incorporating structural and population information improves variant effect predictions. In addition, we have seen that you can calculate deletions as well with ESCOT, but you have to try different important schemes for deletions. It is not the same important scheme like max score. After this point, I would like to show you how basically you can test it with your own proteins, all possible variant effects. We have a web server, and I tried to put the QR code here. And basically it's prescott.lcqv.upnt.fr. And when you go to this server, you will see 3 main sections. One is on the left-hand side, the blue one, ESCOT web server. In the middle, we have PRESCOTT web server that you can incorporate population information. And the third part is the database. I will introduce them. So when you click on the blue part and you will come to this page, you will need 2 inputs. The first one is a multiple sequence alignment file in FASTA format. The second one is a PDB structure. And we have a third optional, a mutation file. For multiple sequence alignment files, there are some requirements. For example, your query should not contain any gaps. Typically, when you do a multiple sequence alignment with MAFT or any other multiple sequence alignment program, it can introduce gaps. You have to remove them with your favorite sequence editor like Yuzuncu, Jalview, or any other editor, or with a Python script, basically. And the second input is a PDB file. Luckily, we are at a really, really fantastic age. You can produce your own PDB files very easily. For this part, especially if you have a sequence, just sequence, my recommendation would be to go to CollabFold and generate both a A3M file and PDB file in one shot in CollabFold. And then you can convert your A3M file to a FASTA format with HHSuite or any other tool. And when you remove the sequences, you can upload them. This server, as I said, will produce, if you don't add the third option, will produce all possible mutational landscape of a protein, which I will show in a short time. Now we have a database as well. The database contains about 19,000 human proteins. And when you go to the green part, you will come to this page. Here, we have a database of 19,000 proteins, and you can search your protein with UniProt ID or with gene name. But my recommendation would be to check it with gene name, check it with UniProt ID, because gene names can be multiple for a protein. Here, I select as an example the most famous protein, TP53, tumor suppressor protein 53. So And when you say submit, you will come to this page. Here, the color red indicates impact of variant is high, and blue indicates the color of indicates that the impact is very low. You can move it to the left and right hand side to see details, and it's an interactive map for your information. When you go over the mutant, you will see the numbers as well as the color. Our source code is also open, and it's creative. It's a Creative Commons license. But as we all know, academic code is ugly typically. So if you want to try multiple proteins, my recommendation would be to go to our Docker image, and you can run it locally on Windows, on Linux, or on a Mac. In this way, you can test hundreds of proteins, but sometimes, you know, on a web server, it's difficult to do it in a batch mode, I would say. So I would like to finish this talk by thanking, of course, our grant sponsor, ANR. ANR is equivalent of NSF in the US or TÜBİTAK in Turkey. And I would like to thank my mentor, Alessandra Carbone. And I would like to Thomas Henry. He was doing the experimental side of this project, and he really inspired us a lot. He made us realize the experimental and clinical aspects of our research. I would like to thank Laurent David. He did the web server part, and it's great, I think, to have a web server to give everybody an opportunity to try the server. I would like to thank Jansi. Jansi is a supercomputing infrastructure management center of France. And of course, I would like to thank Jansi. Jansi is the GPU-based supercomputer in France. They provide us resources for entire human proteome-wide calculations. And I would like to finish by thanking you both for your invitation and for your kind welcome. If you have some questions, I can take it.
Thank you for your precious talk, dear professor. I think we can take the questions now.
I think I have to stop sharing the screen, right?
You stopped.
Oh, okay. Okay, now.
Okay, maybe I can go first. Thank you so much for your, it was a very interesting talk and very impressing, very, I think it's gonna open up different ideas, because this is a very interesting problem, right? Like, I mean, we are all looking at it and never have an answer, definite answer, even though now we can predict 3D structures, still we cannot understand the effects. I have one question. It is related to calculating the distance. You were mentioning about calculating the I think the max score. While you are calculating the max score, there is this evolutionary distance calculation.
Yes.
And
yes.
I was wondering whether there is a different weights for, like, for example, amino acids have different psychochemical properties, right? If
Yes.
I am changing, for example, an aromatic to something, I don't know, a basic or something like that, it's are you counting this or not?
Yes, implicitly, but not explicitly. Implicitly in the following sense, the PC parameter in that equation in max score, it's taking it into account, but there is not a guarantee that it will select TZ plus PC part or TZ plus CV part. If, for example, it's highly conserved and if the highly conserved and buried, it will select the structural part. But if it's not highly buried, it will select the PC part. Therefore, yes, it is considered, but it's implicitly considered. And there's not a guarantee that it will always pick up that component.
Okay, I see. So everything is actually more relying on the evolutionary conservation.
Both evolutionary conservation and PCCV, but really, I mean, how can I say? Sometimes an amino acid can be important because it's conserved. Sometimes it can be due to your, for example, your MSA is not sampling enough, but it's a buried amino acid, which means typically if it is buried, it is an important amino acid. Sometimes it is important, but it's at the surface because it's connecting to, it's interacting with another protein, it's interacting with DNA, it's interacting with ligand, etc. So this kind of approach, I mean, like a max score, gave us a chance to select it based on the weights they produce. So it is considered, as I said, but it's implicit, not explicit.
Okay, thank you so much. Another question. I also have
Sure.
another question. So, in this method, we are also relying on the atomic density, I guess, right? So
Yes.
the structural information.
Yes.
So the disordered proteins, for example, we have absolutely no idea and cannot use this.
Mm-hmm. Actually, unfortunately, for some questions we have yes and no answer, but for this kind of questions we do not. You can use it because in the bioarchive paper and hopefully in the to be published, I would say, paper, we investigated whether ESCOT can predict mutations in disordered segments of a protein. And ESCOT performance was quite good. For some proteins, better than AlphaMissense. For some proteins, at the same level. To predict disordered parts also. For disordered proteins, the CV component will be less, but that doesn't mean that you cannot predict it. Evolutionary conservation still provides information. And in our bioarchive paper, you can see an analysis for spastin 1. It's also a human gene. I think it plays a role in some diseases, but I am not an expert, definitely on the clinical side. So it can still predict for even disordered proteins. It can predict variant effects.
Oh, very interesting. Thank you so much. I'll try myself. Thank you.
Okay. You're welcome. Any other questions?
I think, and it is all.
Okay.
For the question side.
Okay.
So I do apologize. I attended like the last minutes because I learned it in the last minute. But I just quickly looked at your paper in bioarchive.
Okay.
So actually, my question is about to understand what you did. As far as I understand, that you basically use like different species in protein sequencing and try to predict the consequence of the mutations. So and then there is like the other different tools. I couldn't check what. I'm sure you probably compared the other tools. But what you added on top of the current tools? Why should we use that? I'm a clinical bioinformatics and bioinformatician. Why my question is, why should I use that?
Because our results are accurate.
Okay. Sorry, I do apologize. I just
No. Okay, joke aside. Okay, now, okay, from the clinical side, I can tell you this. First of all, computational methods are currently, according to ACMG guidelines, currently supporting information. Okay.
Yeah.
So none of the computational methods are right now in use for clinical decision making. But considering the pace of change in computational fields, in AI, in machine learning, deep learning, I am almost sure that it will be used in a very, near time. Like, I don't know, maybe 5 years, maybe 2 years, maybe 1 year later. So that is the first from the, let's say, clinical perspective.
Yeah.
And these methods are improving gradually. I mean, like, we are, of course, comparing our results with state-of-the-art methods, and our results for human predictions especially are one of the best, I would say, if not the best. Right now, AlphaMissense, which has been published last year, AlphaMissense, I mean, like, is widely used. They have a website. You can reach their data through their web server, etc. But I mean, we produce same level of accuracy with AlphaMissense. Another thing in AlphaMissense, it is trained on population data. In our case, there's not any training. Therefore, we claim that, okay, there's not any bias introduced by this kind of, I mean, like, training on the data.
Yeah.
So, I mean, that would be our advantage. Even, I mean, like, the PRESCOTT part does not consider any population data.
Yeah.
And despite this fact, without considering any population data, we produce same level of accuracy. So this shows, I mean, shows us two things. Okay, there is still room, actually, a lot of room to improve, whether based on deep learning or just, let's say, classical biophysical methods, to improve this kind of variant effect prediction methods. And if it is good, I mean, one should definitely, the clinicians also should definitely take a look, because unfortunately, okay, we know only about 1 million, 2 million variants in ClinVar, but we have 71 million unknown variants. We have to utilize computational methods. You have to take a look at this from clinicians' perspective. Okay, what does it say? Because with this speed of experimental and clinical evaluation, it's impossible to get to the 71 million.
Yeah. As far as I'm saying, we're focusing on the missense variants.
Yes, exactly.
Yeah. So I'm mostly working on the structural variation, so I'm just aware that apart. But as far as I remember, that my colleagues are using like mostly Revel, because I think the users most. So I do apologize again. I haven't checked your paper and asking these questions. But
No. I mean, feel free. I'm happy.
Also, I have a student, and then working on different tools and comparing the loss of function and gain of function score and with the truth set. So I'm just thinking of add that tool and then compare with the others. So, but I need a justification to just present to my PI and add that tool.
So, I mean, like, one thing that I can note about gain of function part, actually, we compared the gain of function for about 13 proteins, gain of function predictions.
Yeah.
And our tool is better in predicting gain of function, pathogenic gain of function mutations, I would say.
Yeah.
So it's better than AlphaMissense. And that's also another advantage of PRESCOTT approach, I would say. But for Revel, if I am not wrong, okay, I'm not 100% sure, but Revel considers scores of many other methods for training. That's what I remember. And it's a supervised learning, if I am not wrong. So unsupervised methods and methods like ours, which are not based on training at all, I think they provide much more valuable information in this sense, because there's not any training involved. So it shows us, okay, we can still develop our current methods further with standard biophysical approaches like physical-chemical features and epistatic effects, etc., or structural information.
Yeah, as you said, it's always lots of room to improve, and that's a great work. Thank you, and thank you for answering questions.
Thank you. Thank you also, Seray.
I think it is the end of our webinar session. Thank you for joining. So especially thank you for the precious talk, dear professor.
Thanks a lot for your invitation.
And close the meeting, I think.
Okay, have a great day.
Have a good day.